Hello, children. We shall now continue with the next topic of the chapter transpiration. Remember, this is very important topic. Photometer. What is a photometer? A device which measures the rate of water intake by the plant. And this water intake is almost equal to the amount of water lost through transpiration. Did you get it clearly? Water intake is equal to the water lost through transpiration. In the previous class, I had told you that although the plant is absorbing water continuously, it only requires 2% for its physiological activities, and the rest goes out to the atmosphere as water vapor. Gannon's photometer. Can you see this diagram? Observe it carefully. You have a twig of a suitable plant. You see here there are no roots. The entire apparatus is filled with water. This is the reservoir which is filled with water. There is a graduated capillary tube which is dipping in a beaker containing water. An air bubble has to be introduced. How? This is done by lifting the bent capillary tube above the colored water so that air may be sucked in due to a suction pull and is again dipped in water. This is lifted up above the colored water and an air bubble is sucked in and is again put in water. So an air bubble is introduced here. As transpiration occurs, you see the water is lost from the twig and a suction force pulls the water from the beaker and the air bubble in the capillary tube moves along. The movement of the air bubble would give us the volume of water lost in a given time. The air bubble can be brought back to the original position by releasing water from the reservoir into the capillary tube by opening the stopcock. So you see here, your air bubble has moved here. Now when it starts moving, as the water is lost by transpiration, it reaches here. Now to push the air bubble back to the original place, we will open this stopcock of the reservoir and the water will go along with it. The air bubble will go back to the original position. Again, for the second time, you can measure the volume of water lost. Is it clear till here? Remember, photometers measure the water uptake by the plant. The photometer must be made watertight and the twig must be cut obliquely underwater such that no air bubble will enter into the twig. Otherwise, absorption of water will not occur. Limitations in the use of a photometer. Introducing the air bubble is not easy. The twig may only remain alive for some time, and any changes in the outside air temperature may affect the movement of the air bubble. Again, I repeat, pay attention carefully, children. What is a photometer? A device which measures the water intake by the plant, which is equal to the amount of water lost by the plant during transpiration. You understand this experiment clearly, and after that you will be doing this question in your notebook. You will do question number 4 in your notebook after understanding the experiment clearly. Kinds of transpiration. There are basically 3 kinds of transpiration: stomatal transpiration, cuticular transpiration, and lenticular transpiration. The major part of transpiration occurs through stomata. I will stop here. Next class, we will discuss the 3 kinds of transpiration. Dear children, I know it's a tough time to understand all these things, but I expect you to do at least 70% of the reading daily. Do your work nicely, neatly. All the best. Take care.